Welcome back, guys. Today in pathology, we are going to discuss this very important topic of cell death. Okay, so let's start the discussion of cell death. First topic we are going to discuss in cell death is necrosis. Necrosis. Necrosis is definition is the pattern of cell death which is always pathological, the pattern of cell death which is always pathological. It's also called accidental cell death. Okay, cell death occurs during irreversible cell injury. When the cell is poised to be dead, there is nothing can be done to save the cell. So this is first part we are going to discuss is necrosis. Okay, so we want to see the mechanism, how it occurs. Okay, there are two ways. Okay, first is denaturation of protein or enzymatic digestion of cells. Digestion of cells. This leads to damage to the plasma membrane. Okay, damage to the plasma membrane, which leads to cellular contents being leak out of the cell. The plasma membrane does not remain intact, it is damaged, cellular contents leak out of the cell. This elicits an inflammatory reaction. Okay, this elicits an inflammatory reactions. This is the short and sweet mechanism of necrosis. Okay, there is damage to the plasma membrane and inflammatory reaction. These two are important features. Okay, now we want to see types of necrosis. Types. Okay, first is coagulative necrosis. Okay, this is the most common type of necrosis. Okay, most common type of necrosis occurs in all solid organs except brain. It occurs in all solid organs. For example, in liver, heart, spleen, kidney. Okay, remember this heart is the most common organ to be affected in coagulative necrosis. Which heart, also liver, spleen and kidney along with heart are also affected. It occurs in all solid organs except brain. Okay, heart is the most common organ to be affected in coagulative necrosis. Now there is a term called infarct. Must have heard it, myocardial infarction. So it's called an infarct. What is this infarct? This is localized area of coagulative necrosis. Okay, localized area of coagulative necrosis. Coagulative necrosis can be considered a type of dry gangrene. Okay, can be considered a type of dry gangrene. These all important points are given in Robbins, so I am saying this. This can be considered a type of dry gangrene. Okay, is a typical feature called Zenker's degeneration. Zenker's degeneration. What is this? It's a special type of coagulative necrosis. Coagulative necrosis seen in typhoid. And it affects rectus abdominis. Rectus abdominis muscle in the abdomen, rectus abdominis, or skeletal muscle. Okay, this is Zenker's degeneration. Special type of coagulative necrosis seen in typhoid, affects the rectus abdominis or skeletal muscle. Okay. Now, what are the microscopical features, H&D features of coagulative necrosis? First is the cell outlines are preserved. Okay, cell outlines are preserved. Second is densely eosinophilic appearance. Okay. Why? Due to loss of cytoplasmic RNA. Okay. There is more glassy appearance. Okay, due to loss of glycogen. Very important feature, more glassy appearance due to loss of glycogen. Okay. Lastly, there is moth-eaten appearance. Moth-eaten appearance. The appearance will be like eaten by a moth. Okay. Why? Due to lysosomal digestion of organelles. Lysosomal digestion of organelles. This leads to this moth-eaten appearance. Moth-eaten appearance, glassy appearance due to glycogen loss, densely eosinophilic appearance due to loss of cytoplasmic RNA. Cell outlines are preserved. These are some of the keywords which could be given in exams. So these are the microscopic hematoxylin and eosin. H&E is hematoxylin and eosin features of coagulative necrosis. Okay. Next type of necrosis is liquefactive necrosis. Liquefactive necrosis. Okay. It is also called, remember, colliquative necrosis. Okay. So Robin says that this is mostly due to enzymatic digestion of cells. Mostly due to enzymatic digestion of cells. For example, in brain injury, in abscesses, in fungal infections. Okay, fungal infections. Remember, coagulative necrosis occurs in all cellular organs except brain. So here in brain injury, this occurs, liquefactive necrosis or colliquative necrosis. So features are cell outlines are not preserved. Cell outlines are not preserved. This can be considered a type of wet gangrene. Robin says it's a type of wet gangrene, which liquefactive necrosis. Coagulative was dry gangrene. Okay. Liquefactive is wet gangrene. Liquefactive occurs mainly in brain injury and in fungal and abscesses, fungal infections and abscesses. Remember, brain is for liquefactive. Coagulative was for most commonly heart and also spleen, kidney, liver. Okay. Then the next necrosis we want to see, very important, caseous necrosis. Okay, caseous necrosis. This caseous word means cheese like appearance. Okay, cheese like appearance. It occurs in, remember, very important, TB infections, tuberculosis infections, very important, single most important example, tuberculosis infection. Then in fungal infections such as histoplasmosis or coccidioidomyosis. Histoplasmosis or coccidioidomyosis, these fungal infections, and also in tuberculosis infection, these caseous necrosis occurs, which pattern is some cheese-like appearance. Okay, this caseous necrosis is a combination of coagulative and liquefactive necrosis. Liquefactive necrosis. Caseous necrosis is a combination of coagulative and liquefactive necrosis. So why this cheese-like appearance occurs in this necrosis? Because the mycolic acid which is present in the cell wall of the bacillus that causes tuberculosis, the mycolic acid, it reacts with the calcium present in the cell walls to form this cheese-like patterns or cheese-like appearance. Okay, so this was caseous necrosis. Single most example, tuberculosis infection. Remember, it's a combination of coagulative and liquefactive necrosis. Fourth one is fat necrosis. Okay, fat necrosis. Fat necrosis is of two types. Okay, first is traumatic fat necrosis. Okay, traumatic fat necrosis is occurs in breast. Okay, and then next type is enzymatic. This enzymatic fat necrosis. Okay, this occurs in momentum, mesentery, and pancreas. Okay, momentum, mesentery, and pancreas. How this fat necrosis occurs, we are going to see. So, as an example, pathology. For example, this is an injury to the liver. This is the liver. It is an injury. So, occurs an injury. This releases the lipase enzyme. Okay, Robbins only mentioned this lipase enzyme. This releases the lipase enzyme. This lipase enzyme, digests fat and leads to release of free fatty acids. Okay, release of free fatty acids. This free fatty acids combines with calcium. This leads to saponification of fat. Combining of calcium with free fatty acids is called saponification of fat. Very, very important. This leads to, remember, chalky white appearance. Okay, chalky white appearance. Don't confuse with the cheese-like appearance. Cheese is also white-like appearance of caseous necrosis, and chalky white appearance of fat necrosis. These two are different when you see the images in the books. you can understand. So this is how fat necrosis actually occurs. Remember this important enzyme lipase leads to fatty acid when combines with calcium leads to saponification of the fat. This leads to chalky white appearance. Okay. The next step of necrosis we are going to see is the fifth one, fibrinoid necrosis. Okay, fibrinoid necrosis. A special type of necrosis that occurs in type 2 or 3, type 2 or 3 hypersensitive reactions. Type 2 or 3 hypersensitive reactions. Here there are antigen antibody combination and immune complexes. Okay, the hypersensitive reactions are immune reactions. There occurs antigen antibody combination and immune complexes. These gets deposited in vessel walls. This gets deposited in vessel walls. Very important. This is the mechanism of fibrinoid necrosis. Immune complexes and antigen antibody combination gets deposited in vessel walls. This has a fibrin-like appearance. It has a fibrin-like appearance. That's why it's called fibrinoid necrosis. Okay. For example, here we have in rheumatic heart disease. Here we find these Ascoff's nodules. These are also seen in malignant hypertension. And hypertension causes hypersensitive reactions. Also in polyarthritis nodosa. In short, PAN, polyarthritis nodosa, malignant hypertension, rheumatic heart disease. This fibrinoid necrosis is type 2 or 3 hypersensitive reactions. The immune complexes gets deposited in the vessel walls as a fibrin-like appearance. Okay. So this was all about discussion of necrosis. Very important discussion. 4 to 5 types of necrosis. All are important. How they look under microscope, under hemotoxylin eosin stain, they are also important. Next type of cell death we are going to see is very important topic for discussion, apoptosis. Apoptosis. This apoptosis is called genetically programmed cell death. Apoptosis is genetically programmed cell death. Necrosis was accidental cell death. Okay, this apoptosis is genetically programmed cell death. It generally means falling off. Okay, this means falling off. Remember, this is usually a single cell death. Necrosis occurs multiple cell deaths, but apoptosis is usually a single cell death. Okay, so apoptosis is a mechanism to eliminate the unwanted cells. It is a mechanism to eliminate the unwanted cells. Okay. This apoptosis can be both, okay, physiological and pathological. Physiological and pathological. Okay. Physiological apoptosis examples we are going to see. First example is during embryogenesis or organogenesis. Embryogenesis or organogenesis. Next example is upon hormone withdrawal, there is involution of hormone dependent tissue. Hormone dependent tissue. Upon hormone withdrawal, there is involution of hormone dependent tissue. Okay. Another example is endometrial shedding. Endometrial shedding during menstrual cycle. During menstrual cycle, endometrial shedding is a physiological apoptosis. Then there is death of harmful lymphocytes and cells whose lifespan is completed. Death of harmful lymphocytes and cells whose lifespan is completed. Okay, so these are some of the examples of physiological apoptosis. Pathological apoptosis occurs during DNA damage in misfolded protein diseases. Okay, yeah. Misfolded protein disease, pathological apoptosis occurs. Okay, then in infections like hepatitis B, here occurs a characteristic body called councilman bodies. Okay, infections like hepatitis B in pathological apoptosis due to DNA damage, also due to misfolded protein diseases. Okay, this in physiological and pathological apoptosis. The examples we are going to see the morphological features. Okay, the morphological features. First is the cell size reduces. This is the earliest morphological feature. Okay, next one is the plasma membrane is intact. Okay, as the plasma membrane is intact, there is no inflammation. Inflammation is absent. Number 4 is chromatin condensation. This is the, remember, most characteristic feature of apoptosis. Most characteristic feature of apoptosis is chromatin condensation. The earliest morphological feature, remember, was cell size reduction. The fifth is, remember, formation of cell membrane Mbbs. Okay, in this capital, cell membrane Mbbs. So what are these Mbbs? See, suppose this is this cell, the membrane. Okay, These membranes actually fall out here, 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 here and here. Okay, membranes actually fall out in pieces. Okay, blebs, they fall off and lead to formation of apoptotic bodies. Apoptotic bodies, formation of apoptotic bodies. Okay, now these apoptotic bodies composed of apoptotic bodies are composed of membrane bound and contains organelles. Contains organelles along with nuclear chromatin. Okay, as we have discussed above, chromatin condensation is the most characteristic feature of apoptosis. These apoptotic bodies, these blebs fall off and form apoptotic bodies which are composed of membrane bound and contain organelles, also the nuclear chromatin. Okay, these are the, remember, eosinophilic bodies. Robin says these are eosinophilic bodies, which these are apoptotic bodies are eosinophilic bodies in formation of blebs, cell membrane blebs. These are the features. Okay, physiomorphological features. Next we are going to see mechanism, the all important mechanism of apoptosis. Mechanism of apoptosis, very very important. Listen carefully. It occurs in 3 phases. Okay, it occurs in 3 phases. First is the initiation phase. Next is the execution phase. Okay, and finally there is removal of apoptotic bodies. The apoptotic bodies that are formed, the removal in the final third step. Initiation phase, this execution phase, and this removal of apoptotic bodies. Before we proceed to the process, there are 2 important enzymes in apoptosis. 2 important enzymes in apoptosis that requires discussion. Okay, first is the caspase enzyme. Okay, caspase enzyme. This caspase enzyme, remember, contains cysteine. And they cleaves near the aspartic acid residue. Aspartic acid residue. Okay, they contain cysteine and they cleaves the near the aspartic acid residue. Okay, these are 2 types. Okay, first is the initiation caspase. Caspase required for initiation phase. Okay, these are, remember, caspase 8, 9 and 10. Initiation caspase, caspase 8, 9, 10, and execution caspases. Execution caspases. These are caspase, remember, 3, 6 and 7. Out of 3, 6, 7, execution caspase, this caspase 3 is the most important one. Very very important fact to remember. Initiation caspase and execution caspase. Second enzyme is our endonuclease. Endonuclease. You know, endonuclease causes DNA breakdown. This causes all important chromatin condensation. The most characteristic morphological feature of apoptosis, protein condensation, is due to endonuclease. Why? It causes DNA breakdown. So caspase and endonuclease. Initiation caspases, execution caspases. Remember these names, we will look at them later. Now, after enzymes, we have some regulators of apoptosis. Regulators of apoptosis. Here we have three factors. First are the pro-apoptotic factors. Okay, pro-apoptotic factors. These are two. Remember, BAX and BAC. BAX and BAK. BAX and BAC. Okay. Next is the anti-apoptotic factors. Anti-apoptotic factors. Remember, these are the BCL2 family. Okay. BCL2 family. What are they? They are BCL2, BCLXL and MCL1. BCL2, BCLXL and MCL1. BCL2 family are anti-apoptotic. That does not allow apoptosis to take place. Pro-apoptotic are BAX and BAC. They allow apoptosis to form. The third group of regulators are called regulated initiators. The regulated initiators of apoptosis. Okay, these are called, remember, stress sensors. Regulated initiators of apoptosis, they are called stress sensors. These are five. What they are? BIM, BID, BAD, NOXA and PUMA. Okay, BIM, BID, BAD, NOXA and PUMA. These are the regulated initiators of apoptosis, also called stress sensors. Pro-apoptotic factors, anti-apoptotic factors and regulated initiators of apoptosis. These are the regulators of apoptosis. So finally, see the mechanism. The first is initiation phase. Okay, initiation phase. Initiation phase is again of two subtypes. First is intrinsic pathway. Pathway is intrinsic. Okay, this is also called mitochondrial pathway. Mitochondrial pathway occurs in 90% of cases. Mitochondrial pathway, the intrinsic pathway, in 90% of the cases. Next is the extrinsic pathway. Obviously, extrinsic pathway. This is called, remember, death receptor mediated pathway. Death receptor mediated pathway. What is this death receptor? We will see further. The extrinsic pathway, death receptor pathway. It is in 10% of cases. Not much prevalent, not much seen. Majority 90% of the cases are the mitochondrial pathway of the intrinsic pathway. So the most common organelle affected in apoptosis is, always remember, mitochondria. Okay. Most common organelle affected in apoptosis is mitochondria. That is why in 90% of the cases it is mitochondrial pathway involved. Okay, so we will discuss one by one. First is the intrinsic pathway. Intrinsic pathway. Okay, the mitochondrial pathway. how mitochondria is involved we are going to see now. Okay, so we know mitochondria have, it's a double membrane organelle. There is this inner membrane, and there is this outer membrane. Okay, inside this inner membrane, here present is this called cytochrome C. You know cytochrome C, the fourth complex of electron transport chain. Inside this inner membrane, it contains this cytochrome C. Okay, now this outer membrane, okay, here the outer membrane permeability is maintained by BCl2 family. Remember the BCl2 family? So here the anti-apoptotic factors. Okay, this outer mitochondrial membrane permeability. Remember, outer membrane permeability is maintained by BCl2 family of proteins. The proteins, BCl family 2 of proteins. Okay, BCl2 are anti-apoptotic. Okay, so the cytochrome C is present inside the inner membrane, inside the mitochondria. Okay, so there occurs the signals of apoptosis. Certain things happen which leads to generation of signals for apoptosis, such as DNA damage, viral infection, okay, viral infection, radiation, and lack of growth signals. Radiation and lack of growth signals. Okay, these all factors leads to, remember, activation of stress sensors. You know the stress sensors, Mbbs, BAD, NOx and PIMA. These are regulated initiators of apoptosis. The activation of stress sensors. Okay, this activation of stress sensor leads to activation of pro-apoptotic factors, BAX and BAC. These are pro-apoptotic, BAX and BAC. Okay, now these BAX and BAC, see here, this is the inner membrane, this is the outer membrane. This BAX and BAC, they do what? They form this channel. Okay, this channel is called BAC channel from inner membrane along through the outer mitochondrial membrane. Okay, BAC channel. Okay, so this BAC channel is for release of cytochrome C enzyme. This BAC channel is for release of BAC channel. Release of cytochrome C enzyme. Okay, then there is release of this cytochrome C enzyme to the outside. Cytochrome C gets released outside. This cytochrome C combines with APOP1. It is APOP1, apoptotic factor 1. Cytochrome C combines with APOP1, apoptotic factor 1, to form, remember, all important apoptosome. Okay, APOP1, apoptotic factor 1, combining cytochrome C is get released from inside the mitochondria is the apoptosome. As long as cytochrome C is inside the mitochondria, there is no signals for apoptosis. Once the cytochrome C gets released through this BAC channel, pro-apoptotic factors apoptosis is inevitable. Formation of apoptosome then activates caspase 9. Remember, these are the initiator caspase. What were the initiator caspases? Caspase 8, 9 and 10. This activation of caspase 9 activates caspase 3, 6, 7. What were these 3, 6, 7? Executional caspase. Executional caspase. This leads to apoptosis. Cell. Activation of caspase 9, initiator caspase, leads to activation of caspase 3, 6 and 7, executional caspase, leads to apoptosis. Okay, this was the whole gist of intrinsic or mitochondrial pathway. It all starts with cytochrome C being held inside mitochondrial membrane, which gets released through this back channel, pro-apoptotic channels, and activation of caspases, which discussed before, important enzymes leading to apoptosis. Now there are inhibitors, some inhibitors of intrinsic pathway. There is IAP, inhibitor of apoptosis. This is the inhibitor of intrinsic pathway. Intrinsic pathway. Then there is SMAC and Diablo. SMAC and Diablo proteins, these are they inhibit the inhibitor of intrinsic pathway. Intrinsic inhibit the inhibitor. What is the inhibitor of intrinsic pathway? IAP protein. SMAC and Diablo inhibit the IAP, inhibitor of intrinsic pathway. These two facts are given in Robbins, so remember them. Okay, this was all about our intrinsic pathway. Now we are going to see extrinsic pathway. Extrinsic pathway. This was the death receptor mediated pathway, occurred in 10% of the cases. To see how these death receptors or death domains are found. Okay. Suppose, take this example. This is a T lymphocyte. Okay, it's a T lymphocyte. Here it has 3 ligands called, remember, Fas ligand. Okay, so Fas ligand. Here in the cell. Okay, it's the cell. Okay, there was 4 domains. Okay, 4 domains in the cell that needs to undergo apoptosis. These 4 are called death domains or death receptors, which, as we can say, these death domains or receptors acts as the receptor for this Fas ligand. These Fas ligand, they join with the death domains through, remember, this element. This element is our Fas. Okay, through Fas ligand in the T lymphocyte combines with the death domains in the cell that needs to undergo apoptosis. Okay, then these two combines, Fas ligand and Fas combine. Okay, now I'm going to see right here. This Fas ligand and Fas combines to generate signal. Okay, signal is generated. Okay, next what happens is 4 death domains. 4 death domains comes together. Seen above, the 4 death domains that come together to form, remember, FADD, but it stands for Fas associated death domains. Okay, fast associated death domains are found. FADD is found when 4 death domains come together. These 4 death domains in the cell comes together to form FADD, fast associated death domain. After this, it's a very simple. It then activates pro caspase 8 to caspase 8. Okay, pro caspase 8 is converted to caspase 8. Remember, this was our initiator caspase. Okay, in intrinsic pathway it was caspase 9. Okay, in extrinsic pathway it was caspase 8. So very important difference, remember, initiator caspase, which leads to further activation of caspase 3, 6 and 7. Okay, this we know these are the executional caspase. Very simple. And finally, which will lead to apoptosis and cell death. Okay, apoptosis and cell death. This is the difference of caspase 8 and caspase 9. Caspase 8 in extrinsic pathway and caspase 9 in intrinsic pathway. Caspase 3, 6, 7 activation, later execution of caspases leads to apoptosis of cell and cell death. This is the difference between intrinsic and extrinsic pathway. Again, the T lymphocytes, there are fast ligands. In the cell, there are 4 death domains. They combine through fast. Okay, the fast ligand and fast, they combine to generate the signal for apoptosis, which leads to 4 death domains in the cell to come together to form fast associated death domain. Then it leads to caspase 8 activation, caspase 3, 6, 7 activation and apoptosis. A bit complex and long pathway, but once we revise and remember, very easy to follow. Okay, this is where the intrinsic and extrinsic pathway. Now I am going to see some inhibitors of extrinsic pathway. There is only one inhibitor of extrinsic pathway, that is FLIP. This FLIP protein is the inhibitor of extrinsic pathway. Extrinsic pathway, FLIP protein, inhibitor of extrinsic pathway. There it was what protein? IAP, inhibitor of apoptosis, which inhibited intrinsic pathway, and SMAC and Diablo were the inhibitors of they inhibited the inhibitors of intrinsic pathway, SMAC and Diablo. Okay, then this is called initiation and execution. 2 phases are over. Okay, and the third part comes removal of dead cells. Okay, removal of dead cells. So how dead cells are removed, we are going to see. So take the example of this cell. This is a normal cell. Okay, in the normal cell, the cell membrane is the inner cell membrane and outer cell membrane. In normal cell, there is this compound called phosphatidylserine. Phosphatidylserine on the inner membrane. Okay, phosphatidylserine, important lipid, important compound lipid, phosphorylated lipid. Phosphatidylserine is on the inner membrane in a normal cell, but in this cell, this is the apoptotic cell. Apoptotic cell, phosphatidylserine comes to the phosphatidylserine, PS I am writing in short, is on outer membrane. Outer membrane. Okay, so phosphatidylserine is on outer membrane. This is called, in the apoptotic cell, this is called, remember, phosphatidylserine flip. Okay, phosphatidylserine flip occurs from inner membrane to outer membrane. Okay. Phosphatidylserine flip occurs, which leads to this compound. This compound is called, remember, annexin 5. Annexin 5, very very important compound. Annexin 5, this is the marker of apoptotic cell. Okay, this is the marker of apoptotic cell. This, it recognizes the phosphatidylserine on outer membrane, okay, and binds to it, okay, and it binds to it, which will lead to phagocytosis of apoptotic cell. Phagocytosis of apoptotic cell. This phagocytosis of apoptotic cell is given a special name. Robbins says this is called efferocytosis. Efferocytosis. Phagocytosis is phagocytosis of apoptotic cell. Remember, annexin 5, very very important. So marker of the apoptotic cell. Recognize the phosphatidylserine on outer membrane and binds to it. Leads to phagocytosis, called efferocytosis. Revolted cells occurs by this phosphatidylserine flip, originating on the inner and then flipping onto the outer membrane. Okay, this was all about our cell death. It was our necrosis, the types of necrosis. In here, the apoptosis, the enzymes involved, physiological and pathological pathways, extrinsic and intrinsic, initiation and execution, and the removal of dead bodies, the inhibitors of both the phases discussed earlier. Very important pathological discussion is cell death. Please do revise them. Thanks for watching, guys. I will catch you in the next one. Good luck.